Hi everyone. I'm Tiffany, and I'm a first-year master's student from the MacArthur Lab at McMaster University in Canada. So I'm really excited today to present some of the work that I've done over the past year using named entity recognition and relationship extraction to mine minimum inhibitory concentration from biomedical text. So let's talk a bit about the biology first. So our lab studies these microscopic organisms called bacteria. Bacteria themselves are found everywhere and can be pathogenic and infectious to humans. So when we have bacterial infections, doctors prescribe us antibiotics. However, that doesn't always work, and that's due to antimicrobial resistance, whereby the bacteria evolve to become resistant to antibiotics. So AMR itself is caused by the overuse and overprescription of antibiotics, and between 2025 to 2050, there's expected to be around 39.1 million deaths directly attributable to AMR. Biologically, AMR is caused by natural resistance, which can be intrinsic or induced. However, it can also occur by being acquired. So bacteria can acquire this genetic material through horizontal gene transfer, typically through plasmids. So a key role in combating AMR is AMR surveillance. This entails the tracking and the detection of AMR genes and pathogens, as well as assessing the spread of AMR across humans, environments, and agriculture. So our lab has created and currently maintains the Comprehensive Antibiotic Resistance Database, also known as CARD. CARD itself is a genomic surveillance resource for AMR, and it's actually the most cited database of its kind. It organizes hand-curated information through ontologies, stores AMR genes, and this arises through a variety of different mechanisms like efflux pumps, limiting the uptake of drugs, so on and so forth. And it even offers software tools such as the Resistance Gene Identifier, or RGI, that allow for the analysis of genomic data. So for my project, we're going to be leveraging the ontological piece and the hand-curated data within the antibiotic resistance ontology, also known as the ARO. So the ARO itself holds several AMR determinants, antibiotics, drug classes, so on and so forth. So we know that the genomic revolution has allowed for researchers to understand the genetic blueprint of an organism. And CARD itself is really good at assessing things on the genotype level. So for example, detecting resistant genes within sequencing data. Now, the issue is, even though we have the sequences, the sequencing information can fall through when there's no phenotypic testing. Because the presence of resistant genes within a sequence does not necessarily mean that resistance will be expressed phenotypically. So to collect this phenotypic information, there's a minimum inhibitory concentration, or the MIC, of an antibiotic. And MIC itself is defined as a concentration at which an antibiotic inhibits bacterial growth. So we can take a bacterial sample from a patient and determine the MIC, and that will tell us if the antibiotic will be susceptible or resistant. So there are several different ways in which people determine MIC. However, it's been standardized through the Clinical Laboratory Standards Institute, CLSI, which is primarily used in North America, as well as a European Committee on Antimicrobial Susceptibility Testing, or EUCAST. So overall, what we want to do is we want to incorporate this MIC information for AMR surveillance, creating a database extension to CARD called CARD MIC, which is my project. So there are a variety of different factors that can impact MIC itself. So the antibiotic, bacterial isolate, the environment, geographical location, the protocol to obtain the MIC, and the site of infection. And we want to capture all of this qualitative information by leveraging natural language processing, or NLP. So throughout the literature, MIC itself can be, or qualitative information can be presented within sentences. So like that bacterial isolate, the sample type, the protocol, and also quantitatively, and that can be through tables. This information can also just appear in sentences. So overall, MIC can appear in both sentences as well as tables. So the reason as to why it's important that we collect and obtain this MIC information is because there's no such database or a data set that holds MIC data right now. By having MIC in CARD, we can monitor the increasing MIC values, as it can be a forewarning for pathogens that may eventually develop resistance. And we can also track the MIC values over time to monitor the emerging resistance and detect trends in antibiotic efficacy. So to obtain the qualitative data, we can extract out terms by leveraging named entity recognition, or NER, to get the geographical location, environment, bacterial isolate, so on and so forth. And we can also leverage relationship extraction to understand what AMR determinants actually confer resistance to which antibiotics. And we can integrate all of that information into CARD. So for my project today, I'm going to describe how I've determined 10,000 papers that actually have MIC information, and how I've used deep learning to obtain this qualitative and quantitative MIC data. Okay, so let's get into the NLP. So identifying the papers associated with MIC information. So the first step was creating a bacterial drug resistance classifier. And I did this by first downloading the PubMed abstracts, as well as full text articles from PubMed Central. So to create and train the classifier, I leveraged medical subject heading terms, or MESH terms. So there were 87,000 bacterial drug resistance abstracts. And I also took a random draw for the negative set. And together that was used to train 16 different classifiers. So each of these classifiers, there's 4 different classifiers with 4 different vectorizers. They were all trained with 5-fold cross-validation. Boxed in orange is the model with the highest F1 score. So using that model, I performed predictions on the 5.3 PMC articles, specifically the ones that had an abstract. So overall, my classifier yielded 105,000 PMC articles that dealt with bacterial drug resistance. So now that I have that information, I asked the question of, do the results of my ML classifier actually agree with human validation? I had the help of 7 undergraduate MacArthur Lab volunteers, and they read through 1,400 abstracts for me. And I used their labels as kind of a reference. And overall, my model was 92.8% accurate. So going forward with these papers, I was happy with that. So I ended up using regular expressions to identify what papers actually had that standardized MIC method. So I searched for the term CLSI UCAST, and then subsequently I searched for MIC. So these 10,000 papers themselves, they can be used for downstream predictions when we perform MIC text extraction for both qualitative and quantitative data. That's what I'll be referring to for this presentation. So named entity recognition. So previous work done in our lab by Armin and Dalitman involved creating CARD EPI, which is a database extension to CARD, which analyzed the transmission and the epidemiology of antimicrobial resistant genes by leveraging NLP. So him and his army of volunteers had hand-curated 8 training data sets for each of these lexicons. So for my project, I leveraged some of these. I took the pre-existing training data sets, and for the AMR determinants coming from the ARO, I referred to that lexicon as ARO ARC because I also wanted to collect antibiotic information. So we know that hand curation itself is extremely tedious and time-consuming. So I wanted to automate this process by using software. Last July, there was a software called Text-to-Term that was released. And Text-to-Term, you can give it an input of your words. And we can map each word to a specific branch or a specific ontology. So I designed my algorithm to go through every single word and label if there was a perfect match from what Text-to-Term had identified, then it would label that antibiotic. So using a mixture of the pre-existing training data sets as well as the one that I had created for antibiotics, I trained a BioBERT model. So for every single lexicon, it's trained on 5-fold cross-validation. And this actually took about a week to train all of these models. And the best performing model is the one with the highest F1 score, outlined in orange. And the predictions themselves were made on the 10,000 MIC papers that I had previously identified. So in total, I had over 1 million annotations coming from all of these different lexicons. So I wanted to evaluate the quality of them. So for this, I had the help of MacArthur Lab expert curators who helped me assess the entities. We essentially look at the context coming from the context of the entity from the sentence, as well as just like logic of if it belongs to that lexicon. So we evaluated 2,400 entities. So 400 entities per lexicon were selected for NER validation. Overall, my model was 95.6% accurate. So using the entities from NER, we can then move into relationship extraction, or RE. So we take the AMR determinants that we determined from the ARO ARG lexicon and the antibiotics from ARO drug. From here, we can determine which ones actually have this confers resistance relationship. And that's what we were looking for to integrate into CARD. So to build my training data set, I had 5,000 sentences involved in this. And so it would be one gene, one drug. And I had the help of MacArthur Lab expert curators again. So creating this training data set was a lot more tedious than I had anticipated. And that's because within the literature, it's not as simple as gene X confers resistance to antibiotic Y. So every single sentence was looked at twice by the curator. So 2 pairs of eyes per sentence, and then a third round if there was disagreements in the labeling. So using those 5,000 sentences, I trained a relationship extraction BioBERT model on 5-fold cross-validation. Again, box in orange is the model with the highest F1 score. To train the model, it actually took about 2 hours, so it wasn't too bad. And using this model, the predictions were made on all of the sentences from entity recognition, which had a gene and a drug in question. In total, I had 10,000 sentences with these positive gene-drug relationships. Again, with the expert curators, we had performed validation on 1,000 of these sentences, 2 rounds again, so 2 pairs of eyes looking at every single sentence. There was disagreements, third round of labeling would occur. And my model was 86.9% accurate in identifying the AMR determinants which confer resistance to whatever antibiotic. So then I wanted to use this Python tool called Uniform Manifold Approximation and Projection, or UMAP. And before I play this 3D visual for those who may not be familiar with it, the closer the points are, that means that they share more of the same resistant gene. So you're going to see a bunch of antibiotics, and the legends are, or in the legend are the different antibiotic drug classes. So the overall goal here was to get a glimpse of how these antibiotics cluster together based on having shared resistant genes. And what we had hypothesized was that antibiotics coming from the same drug class, they should cluster together more because they share more of the same resistant genes. And that's kind of what we see. So you can see in yellow, the beta-lactam antibiotics, they cluster together. And then in the bottom left here in blue, we have the glycopeptide antibiotics, which also cluster together because they have the same resistant genes. However, we can also hover over the data and you can see what resistant genes are associated to whichever antibiotics. And I know that the relationship extraction model itself isn't perfect and there are some like discrepancies. And to fix that, it would just be further filtration methods to ensure more accuracy of the model. So now moving into the quantitative piece, because now we have the NER and the RE data. So we have all of this qualitative information. So we have entities that tell us about the context about the bacterial samples, as well as the relationship extraction. So what genes confer resistance to which drugs. So the actual MIC values now, what we did was we leveraged ChatGPT. I used the GPT Turbo 4.0 model to extract MIC tables from these papers. So my thought process here was I wanted to design a prompt to extract out table data from full text articles. I also wanted, and then I also wanted to make another summary file, which would tell me a list of all of these PubMed Central IDs, labeled a 1 or a 0, telling me if there's an MIC table involved in that paper. So I inputted the 10,000 articles with MIC mentions, and ChatGPT gave me almost 8,000 MIC tables extracted from 6,000 different PMC articles. And to the right there, here's a table that ChatGPT had extracted. And here's how it actually looks in PubMed Central. So at a glance, it looks like everything it's not hallucinating, which is good. The actual table contents are preserved. But that leads me into my future direction. So everything that I've done so far, that was for the MIC tables themselves. I think it's important that we perform human validation to make sure that the actual table data is about MIC, and also that the values within the tables are also not, they're also true. I also want to extract out MIC sentences, and finally integrate all of these data into CARD, a part of the CARD MIC module. So from this talk, there are two main key takeaways that I'd like you to get from it. And it's that phenotypic testing data can be obtained through text mining, and we can use that to guide AMR surveillance. And the second is that throughout this presentation, you've seen that I've performed human validation 3 different times. I think it's extremely important that when we leverage AI, I think it's key that we analyze the data as empirically as we can to ensure that the data set itself is robust. Aside from that, there's so many people that I'd like to thank who've been a part of the project. My undergraduate volunteers, the MacArthur Lab expert curators. It was cool to have my own PI involved. It gave him homework, basically. Our postdocs, staff members, and my fellow graduate students. I'd also like to extend a special thank you to our software developer, Amos, for helping me with all things ChatGPT. And as well as my committee members, Dr. John Stokes, Dr. Jerry Wright, and all past and present MacArthur Lab members. So aside from that, I'd be happy to take any questions. Thanks.
Thank you so much for a nice presentation. Do we have any questions? Yes. Okay.
The table with the very high precision or accuracy, 99%, was that the question was yes or no? Are there those antibiotics? Did these papers talk about antibiotic or not?
For the classifier?
Yeah, the one table with a lot of 99%, like with a very high, one of the first tables.
Oh, okay. I think that was probably,
Like, what was the question?
I think it was this one.
Yes.
Oh, for this one, that was training the NER model. So extracting out the entities.
Whether it is or it is not.
Whether it is like an annotation from the lexicon that I wanted. So like, is it a gene that I want, like an antimicrobial resistant gene that I want? Is it an antibiotic? What environment is it coming from? Hospital, wastewater treatment plant, things like that.
If you have if you have more than that to ask, why don't you ask directly like a large language model all of the questions too?
Oh, like use large language models to extract that information. Yeah, I guess you could do that like through, but so because we want to kind of create this data set that has all of that kind of information. So I just wanted information about the sample themselves. And also previous work in our lab also just used NER and RE. So it was just kind of the consistency of that as well.
Great talk and very impressive results. For your relation extraction, do you have any sense of when it's wrong, kind of what it's getting wrong, kind of any obvious patterns?
Yeah. So what I had noticed and what was really interesting is that when there's multiple genes and multiple drugs in a sentence, right? When we use words like, oh, this gene, this drug, whatever, and there's like a whole list of them. And then we go respectively. Yep. That really messes up the model. It's not good at that. So what I'm planning to do is to make it a lot more accurate is I want to eliminate all of those cases. And yes, we're cutting out a lot of data, but I also want the data set to be of higher quality. I want us to trust the robustness of what my model's extracting. So I'd say that, as well as sentences where people are saying there was lower resistance reported. Lower resistance still means that there's resistance, but the model will misinterpret that.
Thanks.
Yeah.
Thank you. I'm sorry we don't have more time. You can find Tiffany afterwards. So let's give her a hand again.